Greetings, everyone. My name is Davi Araújo Aragão. I am a pharmacy major at Federal University of Ceará, and I am here to present a study called In Silico Study of Molecular Docking of Natural Limonoids Against Main Protease of SARS-CoV-2. So, to begin our presentation, SARS-CoV-2 is a virus that caused the COVID-19 pandemics. It began in late December 2019 in Wuhan, China, and spread worldwide through the years of 2020 and 2021. In this virus replication process, there is an enzyme which is fundamental for its processing. It's called main protease, or MPRO for short. This enzyme is fundamental in the polyprocessing chain and in the virus maturation processes. It has been discovered that there are some substances with the power to inhibit MPRO. These are originally N3 and then a few other drugs that we already have and use for other diseases, which could be baricitinib, remdesivir, azithromycin, and also anacura. These substances are known for inhibiting this enzyme, but the mechanism of such inhibition has yet to be proven, and this paper will be a part of this. So it has also been studied that some limonoids could be potential MPRO inhibitors. Limonoids are natural terpenoids originated from the molecule tiruculol, and they are also known for their antibacterial and antiviral activities. Therefore, it's believed that they could be used against the SARS-CoV-2. So, talking about the methodology of this study, at first, molecular structures were obtained from two different sources. At first, the control drugs, which are remdesivir, azithromycin, baricitinib, and anacura, their structures were obtained from the PubChem database. And as to the tested limonoids, their structures were constructed with the Marvin Sketch software. As to structural optimization, the authors of the paper chose to use potential energy of each binding as a parameter to analyze the possibility of each interaction occurring. And as a pattern for molecular dockings, the tests that had the lower potential energy were also considered the best ones. And for molecular attachment analysis, the software Autodoc Vina was used, in which it was ran 100 cycles with 10 simulations each. So, to begin explaining the results of molecular docking, the control drugs baricitinib, anacura, remdesivir, and azithromycin were able to inhibit MPRO by binding to the same region as N3 does. Limonoids were able to inhibit MPRO by binding to that same spot as the control drugs and N3. And the mean potential energy of each binding was found to be -7 kilocalories per mole. The root mean square division values of all interactions was found to be under 2 angstroms. And a special spotlight to the interactions with the amino acid residue threonine 199, which appears to form several bonds with the limonoids. Now, to discuss the results, we can conclude that the limonoids have an important role in MPRO inhibition, and that they could be used in further studies to assess their efficacy of inhibition. The results of the molecular docking really do prove to us that the limonoids could bind to MPRO, it would be a favorable bond, and that it could it could come to be a drug, medicine, or new models for such molecular target.